In 1953, a groundbreaking discovery shattered our understanding of biology and set the stage for a revolution in science. Two young scientists, James Watson and Francis Crick, unveiled the structure of DNA, describing it as a double helix, a twisted ladder composed of nucleotides. This discovery not only revealed the molecular blueprint of life but also posed questions that would tantalize scientists and laypeople alike for decades to come. However, the journey to this revelation was long and winding, filled with brilliant minds, experiments, and the faint echoes of rivalries. The story of DNA begins as early as the mid-19th century with the work of Friedrich Miescher, who first isolated a substance he called nuclein from white blood cells. This was the first hint that there was something unique within the cell nucleus, something that contained the essence of heredity. But it wasn't until the 20th century that scientists began to understand the organization and function of this remarkable molecule. In the 1940s, researchers like Erwin Chargaff discovered that DNA was made up of 4 nucleotide bases: adenine, A, thymine, T, cytosine, SC, and guanine, G. Chargaff's rules, A pairs with T and C pairs with G, were crucial clues that would help Watson and Crick construct their model of the DNA molecule. Meanwhile, Rosalind Franklin was capturing breathtaking X-ray diffraction images of DNA, which would ultimately provide key insights into its helical structure. In their landmark paper published in the journal Nature, Watson and Crick wrote, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." This statement would launch a new era in genetics and molecular biology. But what did this mean for the future? The implications were monumental. Understanding DNA opened the doors to the world of genetic inheritance, the basis of evolution, and eventually, a whole suite of modern genetic engineering tools. Fast forward to today, and we are on the cusp of being able to manipulate the very essence of life. But with great power comes great responsibility. As we begin to unlock the potential of DNA, ethical questions loom large. Should we edit the genes of unborn children? Can we eradicate diseases through genetic modification? As we look into the future, we must also grapple with the possibilities of unintended consequences. The discovery of DNA was just the beginning. The real journey of genetic exploration was yet to unfold. As we delve deeper, we will explore how this amazing molecule has led to revolutionary advances in medicine, agriculture, and even conservation efforts. But first, we must understand the technology that enabled these achievements, laying the groundwork for some of the most profound changes in our world. What would happen if we could rewrite genetic code? Could we design organisms to meet our needs? As we move forward, the answers might surprise you. Genetic engineering stands as one of the most potent tools in modern biology, a scientific discipline that allows us to directly manipulate an organism's DNA. At its core, genetic engineering involves the use of various techniques to alter the genetic material of an organism, whether by introducing, enhancing, or deleting specific genes. It sounds like science fiction, but the reality is that genetic engineering has become a fundamental aspect of modern science. Central to understanding genetic engineering is the technology known as CRISPR-Cas9. This gene editing tool, discovered in the microbial world, allows scientists to cut DNA at specific locations. Imagine a pair of molecular scissors that can snip out defective genes and replace them with functional ones. Since its introduction in 2012, CRISPR has been heralded as a major leap forward in biotechnology, enabling targeted modifications that were previously impossible. The versatility of CRISPR is astounding. It has been used to engineer crops for greater yield and resistance to pests, developing strains of rice that can thrive in saltwater, and even creating genetically altered mosquitoes to combat the spread of diseases like malaria. The potential applications seem endless. But how does CRISPR work? At its core, the system is based on a natural defense mechanism found in bacteria, which use it to fend off viral attacks. The bacteria retain a memory of past invaders in their DNA, using this information to recognize and cut foreign DNA in future encounters. Researchers adapted this system for use in other organisms by creating a guide RNA that leads the Cas9 protein to the target DNA sequence. When the Cas9 enzyme arrives at its destination, it makes a cut, allowing the cell's natural repair mechanisms to take over. This repair process can be harnessed to introduce new genetic material, effectively allowing scientists to edit the genome at will. But the excitement around CRISPR is matched by caution. The ability to edit genes raises profound ethical questions. Should we use this technology to enhance human traits? What are the implications of creating genetically modified organisms? The scientific community wrestles with the implications of designer babies, where parents could select for traits like intelligence or attractiveness. Such decisions could lead to a new form of inequality based on genetic enhancements. As we explore further, we must ask ourselves, who gets to decide how we use these powerful tools? The answers may determine the future direction of genetic research and its impact on society. In the next chapter, we will delve into the applications of genetic engineering that are already reshaping our world in ways we might not expect. What if the solutions to some of humanity's most pressing challenges lie within our grasp, yet the choices we make today shape our future in unforeseen ways? The application of genetic engineering in agriculture has the potential to revolutionize the way we produce food. As the global population continues to rise, with estimates suggesting it could reach nearly 10 billion by 2050, ensuring food security becomes increasingly critical. One way to address this challenge is through the development of genetically modified (GM) crops. GM crops are engineered to possess desirable traits, such as resistance to pests, herbicides, and environmental stresses like drought. For instance, Bt corn has been modified to express a protein from the bacterium Bacillus thuringiensis, granting it resistance to certain insects. This has not only increased yield but also reduced the need for chemical pesticides, benefiting both farmers and the environment. In the United States alone, approximately 90% of the corn and soybeans grown are genetically modified. These crops have contributed to significant increases in agricultural productivity, but the debate surrounding their safety continues to be a contentious issue. Critics argue that GM crops could have unforeseen ecological consequences, while supporters point to the extensive research demonstrating their safety. Countries around the world have taken various approaches to GM crops. In some regions, such as parts of Africa, farmers are keen to adopt genetically modified varieties that can withstand harsh climates and pests. For example, the development of drought-tolerant maize aims to help farmers in sub-Saharan Africa cope with the impacts of climate change. On the other hand, several countries in Europe maintain stringent regulations against GM crops, driven largely by public perception and concerns over food safety. The story of genetic engineering in agriculture is one of innovation, but also of resistance. For individuals skeptical about GM technology, stories abound of products marketed as natural that may have no real advantages over their engineered counterparts. Interestingly, studies have shown that GM crops can lead to reduced greenhouse gas emissions, as their efficiency allows for decreased land use and less fertilizer application. As we shift gears to examine the environmental impact of agriculture, the role of genetically modified organisms is becoming ever more significant. Will GM crops be the answer to feeding our growing population, or do they pose risks that we cannot afford to ignore? In the next chapter, we will explore how genetic engineering is not only shaping agriculture but also influencing the field of medicine in profound ways. Genetic engineering's impact on medicine is perhaps one of its most hopeful and life-changing applications. The ability to manipulate genetic material has given rise to novel treatments and breakthroughs that were previously unimaginable. From gene therapy to personalized medicine, genetic engineering is reshaping how we approach healthcare. One of the most exciting realms of genetic engineering is gene therapy. This innovative field focuses on treating diseases by directly targeting the underlying genetic causes. For example, in conditions like cystic fibrosis, characterized by a mutation in a single gene, researchers are exploring ways to deliver healthy copies of that gene to patients' cells. Early results from clinical trials have shown promise, offering new hope to patients suffering from genetic disorders. Moreover, the use of CRISPR technology has revolutionized opportunities for genetic repair. Scientists have successfully used CRISPR to alter genes in various organisms, including human cells, to correct mutations responsible for genetic diseases. Trials are underway to assess the effectiveness and safety of CRISPR-based therapies, paving the way for a new era of medicine. However, these advancements are not without ethical implications. The power to edit the human genome raises fundamental questions. Who decides which genes should be changed? What are the potential long-term effects of gene editing in humans? The concept of personalized medicine, tailoring treatment to individual genetic profiles, is fast becoming a reality. By analyzing a patient's genetic information, doctors can select the most effective treatments and predict responses to certain medications. This approach not only enhances the efficacy of treatments but also minimizes potential side effects. The future of medicine lies not only in treating symptoms but also in understanding the very blueprint of our biology. However, the journey of genetic engineering in medicine is fraught with challenges. The cost of gene therapies can be exorbitant, raising concerns about equitable access to these life-saving treatments. Furthermore, the long-term effects of such interventions are still largely unknown, and researchers are working diligently to understand both the benefits and potential risks. As we continue our exploration, it's crucial to remember that with the potential for healing comes the responsibility of careful consideration. How we navigate these ethical waters may very well shape the future of medicine. In the next chapter, we will delve into the intriguing field of genetic engineering and its role in conservation efforts. While the power of genetic engineering is often highlighted in agriculture and medicine, its potential in conservation is equally profound. As species face unprecedented threats from habitat destruction, climate change, and disease, genetic engineering may offer innovative solutions to preserve biodiversity. Let's explore how scientists are leveraging genetic tools to help endangered species and restore ecosystems. One of the most captivating applications is the use of genetic engineering in the conservation of endangered species. For example, the California condor, once on the brink of extinction, has been the focus of extensive conservation efforts, including the use of techniques like artificial insemination and the understanding of genetic diversity within the population. Researchers are looking at how to enhance genetic diversity through genetic management, which can improve the overall health of these birds. Another fascinating case is the use of genetic techniques to combat the spread of diseases affecting wildlife. The use of genetically modified organisms to combat diseases, such as using genetically altered bacteria to control invasive species, has shown promise. The introduction of these engineered solutions raises questions about the balance of ecosystems and the potential ripple effects of introducing GMOs into wild populations. Yet, these innovative solutions may be necessary as traditional conservation methods become less effective. Moreover, genetic engineering may play a crucial role in preserving critically endangered species. Techniques such as cloning and editing genome sequences might allow researchers to resurrect extinct species, an idea that captures the imagination. While projects like de-extinction are still in their infancy, they spur discussions about the implications of such interventions. If we can bring back woolly mammoths, should we? The ethical dilemmas surrounding these initiatives force us to reflect on our responsibility as stewards of the planet. However, the integration of genetic engineering in conservation is not without controversy. The introduction of GMOs into natural ecosystems remains a hotly debated topic. Critics argue that we risk disrupting delicate ecological balances, and caution is needed when deploying such technologies. Nonetheless, proponents argue that the innovative potential of genetic engineering offers a lifeline to species that have little hope of survival otherwise. How can we responsibly harness this powerful technology for the good of our planet? As we approach the conclusion of this exploration into DNA and genetic engineering, we begin to see a pattern. The intersection of innovation and ethics shapes our future. For the next chapter, we will summarize our findings and reflect on the broader implications of our journey into the world of genetic modification, exploring what it means for humanity's future. As the capabilities of genetic engineering expand, so do the ethical implications that accompany them. The prospect of directly altering the genetic makeup of living organisms raises profound questions about morality, responsibility, and the future of life on Earth. Some argue that this technology can lead to unprecedented advancements in health and agriculture, while others warn of unforeseen consequences and ethical dilemmas. In 2015, a landmark meeting was held in Washington, D.C., where scientists and ethicists gathered to discuss gene editing in humans, particularly focusing on CRISPR technology, which allows for precise alterations in the DNA sequence. The potential to eliminate genetic disorders such as cystic fibrosis or Huntington's disease offers hope to millions. However, it also poses the risk of designer babies, children whose traits could be customized for higher intelligence, physical appearance, or athletic ability. This could create a society divided by genetic privilege, raising the question, who gets to decide what constitutes desirable traits? Consider the case of He Jiankui, a Chinese scientist who, in 2018, announced he had created the first genetically edited babies, Lulu and Nana, resistant to HIV. His announcement sparked outrage and led to his imprisonment, illustrating the tension between scientific advancement and ethical boundaries. Moreover, the potential ecological impact cannot be ignored. Genetically engineered organisms released into the wild could disrupt existing ecosystems, leading to unforeseen consequences. A notorious example is the genetically modified Aedes aegypti mosquito, designed to reduce the population of dengue fever carriers. While initial studies showed promise, the long-term effects on ecosystems remain poorly understood. The ethical landscape of genetic engineering is complex and multifaceted, requiring ongoing dialogue among scientists, ethicists, and the public to navigate the potential risks and benefits. As we move forward, it is imperative to consider the broader implications of our capacity to manipulate life itself. Will we empower humanity, or could we be playing God? The debate continues, and as we explore further, we must confront the moral responsibilities that accompany our newfound abilities in genetic engineering. The future of genetic engineering is both thrilling and daunting. As technologies like CRISPR and synthetic biology evolve, they promise to revolutionize fields ranging from healthcare to environmental science. Imagine a world where genetic modifications can not only eradicate genetic diseases but also enhance human capabilities. This is the vision many scientists share as they explore the frontier of human enhancement. Researchers at the Harvard University Weiss Institute are working on programmable gene circuits that can be used to fine-tune the behavior of living organisms. Their innovations could lead to personalized medicine strategies that adapt treatments based on an individual's genetic makeup. Think about patients with cancer. Instead of one-size-fits-all chemotherapy, we could have therapies specifically designed to target the unique genetic mutations of each tumor, optimizing efficacy and minimizing side effects. Furthermore, genetic engineering could transform how we address global challenges, such as food insecurity and climate change. Scientists are experimenting with genetically modified crops that can withstand extreme weather, pests, and diseases. For example, drought-resistant maize developed by researchers in Africa has shown remarkable resilience and increased yields in arid conditions. Another exciting prospect is the rise of gene drives, which use genetic engineering to spread particular genes throughout a population rapidly. These could be utilized for controlling invasive species or eradicating diseases like malaria. However, such power raises critical questions about ecological balance and long-term impacts. As we envision the future, we must also consider the ramifications of genetic inequality. If genetic enhancements become accessible mainly to the wealthy, we risk creating a genetically stratified society. How might this shift societal dynamics and widen the gap between those who can afford enhancements and those who cannot? The future holds incredible potential, but it also demands careful consideration of our actions today. As we stand on the precipice of extraordinary capabilities, we must weigh the ethical implications and societal impacts of our choices as stewards of the genetic fabric of life. As we delve deeper into the application of genetic engineering in natural ecosystems, we encounter both incredible hopes and daunting challenges. The use of genetic engineering in wildlife conservation has emerged as a beacon of hope for many endangered species. For instance, the California condor, once teetering on the brink of extinction, has seen a resurgence thanks to targeted breeding programs and genetic interventions. These initiatives allow conservationists to introduce genetic diversity into the population, bolstering its resilience against disease and environmental change. But the landscape of genetic interventions is not without controversy. Take, for example, the ambitious de-extinction projects aimed at bringing back species like the woolly mammoth. Researchers at Harvard have been working on editing the genes of Asian elephants to create creatures that exhibit traits characteristic of their long-lost relatives. While this initiative ignites the imagination, ethical questions loom large. Will we have the ecological frameworks in place to support reintroduced species? Moreover, the potential for genetic engineering to inadvertently disrupt existing ecosystems cannot be understated. As illustrated by the case of genetically modified fish, there is a clear risk that these engineered organisms could outcompete their wild counterparts, leading to unforeseen consequences in local biodiversity. Furthermore, the reliance on genetic engineering in conservation raises concerns about taking away focus from traditional conservation efforts, the preservation of habitats and ecosystems. Conservationist Roger Braithwaite once wisely remarked, "Nature will find a way, but we cannot assume it will be the way we want." This underscores the gravity of intervention through genetic engineering versus the intrinsic value of natural processes. And yet, genetic engineering holds promise even in combating invasive species. By genetically altering species to dramatically reduce their reproductive output, we could reclaim ecosystems overrun by non-native plants and animals. But as we continue exploring uncharted territories, the question remains, do we have the right to meddle with the very fabric of life? The answer is as complex as the technology itself, full of potential, yet fraught with risks. How we choose to harness this power will define not just the future of conservation but the legacy we leave for generations to come. Throughout the history of genetic engineering, we've witnessed both resounding successes and notable failures that reveal the complexity of this powerful tool. One of the landmark success stories is the development of genetically modified insulin. In the 1970s, scientists successfully inserted human genes into bacteria to produce insulin, revolutionizing diabetes treatment. This innovation not only improved the quality of life for millions but also showcased how genetic engineering can provide tangible solutions to human health challenges. Another fascinating case is that of golden rice, engineered to produce beta-carotene, a precursor to vitamin A. With millions suffering from vitamin A deficiency, golden rice aimed to provide a nutritional boost and prevent blindness and deaths in developing countries. However, the journey has not been without obstacles. The project faced backlash from anti-GMO activists, leading to a significant delay in its introduction. The tensions surrounding golden rice highlight the complex interplay between scientific progress, public opinion, and regulatory hurdles. In stark contrast, consider the failure of genetically modified dogs, aimed at producing hypoallergenic pets. The initiative faced significant challenges not just scientifically but also socially. Pet owners were unwilling to embrace genetically altered companions. These examples illustrate the thin line genetic engineers walk between innovation and public acceptance. As we move forward, it becomes clear that for genetic engineering to truly succeed, it must not only provide scientific advancements but also engage the public in meaningful dialogues. The history of this field demonstrates that transparency, ethical considerations, and education play crucial roles in shaping perceptions and ensuring successful implementation. What are the lessons we can glean from these stories? They reveal the importance of adaptability and the necessity to align scientific endeavors with societal values. It is this alignment that may ultimately dictate the future trajectory of genetic engineering. As we conclude our exploration into the world of DNA and genetic engineering, we must reflect on the profound implications for our collective future. The delicate balance of harnessing genetic technology responsibly lies in our hands. Imagine a world where genetic diseases are eradicated, crops can thrive in inhospitable climates, and where humanity can push the boundaries of its own biology. But this future is fraught with responsibilities, choices, and ethical considerations that we must grapple with. One key takeaway is the importance of stewardship. As we advance in our capabilities, we must ensure that genetic engineering does not become a tool of division but rather a means to elevate humanity as a whole. Education about genetic technologies must become a priority, empowering future generations to understand both the science and the implications of their decisions. Therein lies the challenge. How do we foster a society that embraces innovation while remaining critically aware of its potential consequences? Furthermore, our understanding of genetics is expanding, raising the prospect of not just curing diseases but also enhancing human experiences, biologically, cognitively, and physically. Indeed, the future of genetic engineering often evokes the question of identity. What does it mean to be human in a world where we can edit our very nature? As we navigate these uncharted waters, it is essential to adopt an inclusive approach, engaging diverse voices in discussions around the technology's direction. Ultimately, our genetic future is a tapestry woven from the threads of innovation, ethics, and humanity. As we stand at this crossroads, ready to embrace the possibilities ahead, let us remember to tread carefully and thoughtfully. The choices we make today will echo through time, shaping the biological landscape of tomorrow. The journey of genetic engineering is just beginning, and how we choose to proceed may define the very essence of life itself. In the world of genetics, few advancements have been as revolutionary as CRISPR technology. This gene editing tool, originally discovered in bacteria, has opened a floodgate of possibilities in the realm of genetic engineering. But what exactly is CRISPR, and how does it function? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It serves as a sort of immune system for bacteria, allowing them to fend off viruses. Instead of the traditional methods of genetic manipulation, CRISPR allows scientists to edit genes with unprecedented precision. Developed further by researchers like Jennifer Doudna and Emmanuel Charpentier, CRISPR has transformed into a tool that could be used not just in labs but also in real-world applications. The potential is staggering. Imagine curing genetic diseases, enhancing crop resilience, or even eradicating entire species of pests that threaten agriculture. In 2020, researchers used CRISPR to successfully eliminate a genetic mutation causing sickle cell anemia in human patients. However, with great power comes great responsibility. Ethical dilemmas arise when discussing germline editing, which could alter the DNA of future generations. As research continues to unfold, so does the debate over what it means to edit life itself. Are we playing God, or are we simply harnessing the tools of nature? In the next segment, we will delve deeper into the ethical landscape surrounding CRISPR and its implications on society and the environment. What will the future hold as we tread carefully on this uncharted path? As we explore the cutting-edge landscape of genetic engineering, gene therapy stands out as a beacon of hope for treating previously incurable diseases. Gene therapy involves the introduction, removal, or alteration of genetic material within a patient's cells to treat or prevent disease. This approach has shown promise in targeting genetic disorders such as cystic fibrosis, hemophilia, and certain types of cancer. One notable case involves a young boy named Jaden, who was diagnosed with spinal muscular atrophy, a condition that leads to the degeneration of motor neurons. In 2019, he received a groundbreaking gene therapy called Zolgensma, which replaced the defective gene responsible for his condition. Within weeks, Jaden's progress was remarkable. He was able to lift his head for the first time. Trials for gene therapy have become increasingly common, with several approved therapies now available. However, the journey to acceptance has been filled with hurdles including regulatory scrutiny, cost issues, and the long-term efficacy of these treatments. The average cost of gene therapy can exceed $1 million, leading to questions about accessibility and healthcare equity. As gene therapy continues to evolve, ethical concerns about genetic manipulation and designer babies loom larger than ever. In this next chapter, we will examine how the commercialization of gene therapy intersects with ethical considerations. Can we ensure that these life-saving treatments are available to everyone who needs them, regardless of financial status? Genetic engineering holds immense potential for enhancing biodiversity, but it also poses significant risks to ecological balance. As we manipulate the genetic code of organisms, we are tasked with the delicate duty of understanding how these changes affect entire ecosystems. Take, for instance, the case of genetically modified (GM) crops. Proponents argue that GM crops can withstand pests and diseases, reducing the need for pesticides and leading to healthier ecosystems. On the other hand, critics warn that introducing genetically modified organisms could upset local flora and fauna. A study conducted by the National Academy of Sciences shed light on the impact of GM crops on non-target species, showing that beneficial insects can be affected by Bt crops designed to repel specific pests. This illustrates an important point. While genetic engineering can aid in agricultural productivity, it also necessitates a thorough understanding of ecological interactions. Moreover, the introduction of genetically modified organisms into the wild raises pressing questions. Should we allow genetically engineered species to interact with their wild counterparts? The answer is not straightforward. In certain cases, engineered organisms might become invasive, outcompeting native species and leading to loss of biodiversity. A notable example is the genetically modified fish known as aquabounty salmon, designed to grow faster than its wild counterparts. If this fish were to escape into the wild, it could disrupt ecosystems and alter food chains. So, how do we balance innovation with responsibility? This chapter will delve deeper into conservation efforts and explore how we can embrace genetic engineering while preserving the ecological integrity of our planet. As we consider the consequences of our actions, remember, biodiversity is not just the fabric of life, it is life itself. As we delve deeper into the vast complexities of genetics, we often overlook the microscopic world that significantly influences our genetic makeup, the microbiome. This intricate community of trillions of microorganisms living within and on our bodies plays a crucial role in health and disease. Recent studies show that the gut microbiome, for instance, not only aids digestion but also has profound effects on our immunity, metabolism, and even mental health. It's astonishing to think that these tiny entities outnumber our human cells by a factor of 10, and their genetic material contributes an incredible diversity of genes that far exceeds our own. The concept of the microbiome challenges the traditional view of genetics as solely a human endeavor, suggesting that our genetic expression can be influenced by the microorganisms that we harbor. For example, researchers have found correlations between the composition of gut bacteria and conditions like obesity, diabetes, and even autism spectrum disorders. This leads us to ask, are our genes truly deterministic? Or are they merely players in an elaborate game influenced by an unseen world? Studies have begun to explore how altering the microbiome through diet, probiotics, or even fecal transplants can result in substantial health changes. One landmark study published in Nature in 2013 demonstrated that obesity could be transmitted through microbiota from one organism to another, presenting a revolutionary perspective on obesity and metabolism. These findings suggest that manipulating the microbiome could be a future frontier in combating various lifestyle diseases. For instance, researchers at the University of California, San Diego, have developed a method to engineer bacteria that can produce beneficial compounds in the gut, potentially revolutionizing how we approach nutrition and health management. But, as with all genetic engineering paradigms, this raises ethical questions. How far should we go in modifying our microbiome, and what unforeseen consequences might arise? The microbiome represents a burgeoning frontier in genetic research, where the implications of our genetic engineering efforts extend beyond our own DNA and into the realms of symbiosis, ecology, and communal genetic networks. With each discovery, we are forced to reconsider the essence of individuality in genetic makeup. Are we merely the sum of our genes, or are we also the product of a complex interaction with the unseen microbial world? As we continue to explore the microbiome's mysteries, we must confront the potential of engineered microbiomes as a means to enhance human health and well-being, while also weighing the ethical ramifications of such interventions. What lies ahead in our understanding of this micro-universe is not just science, it's a journey into redefining what it means to be human. In a world grappling with the enormity of climate change, genetic engineering presents itself as a promising tool to combat ecological crises. With predictions indicating that global temperatures could rise by 1.5 to 2 degrees Celsius in the next few decades, scientists are looking to genetic modification as a means to enhance the resilience of various species to climate stresses. For instance, researchers are working on engineering crops that can withstand extreme temperatures, drought, and salinity. A prime example is the development of drought-resistant corn by a biotech company called Pioneer, which has led to increased yields even in the face of water scarcity. Additionally, scientists at the University of Illinois have introduced genes from a plant that thrives in extreme conditions into soybean crops, resulting in plants that can produce more under drought conditions. The potential for such advancements is immense, but they also come with complications. The introduction of genetically modified organisms, GMOs, into ecosystems has sparked debates on biodiversity and ecological balance. When modified crops are introduced to the wild, what happens to the native species? Research is still ongoing regarding the long-term impacts of GMOs, as seen in cases where engineered traits unintentionally spread to wild relatives, potentially disrupting local ecosystems. For instance, the escape of glyphosate-resistant genes into wild populations has sparked concerns and raised questions about the long-term sustainability of such engineering practices. Furthermore, innovators are exploring genetic engineering in other areas such as coral reefs, which are suffering from bleaching due to rising ocean temperatures. Scientists are working on genetically modifying coral species to enhance their resilience to heat and acidification. Initiatives like the Coral Restoration Foundation are employing genetic techniques to restore and rehabilitate coral populations suffering due to climate impacts. The success of these initiatives could redefine conservation strategies and offer new hope for underwater ecosystems. However, as we charge ahead, the ethical implications of playing God in nature cannot be ignored. As we engineer organisms to potentially save others from extinction or enhance agricultural resilience, we must tread carefully. What responsibilities do we bear as stewards of the planet, and how do we reconcile technological advancement with the preservation of natural biodiversity? The intersection of genetic engineering and climate change is a critical area that demands our attention, as it could very well determine the fate of our ecosystems and future generations. Synthetic biology is often seen as the next evolutionary step in the field of genetic engineering. It encompasses the design and construction of new biological parts, devices, and systems, pushing the boundaries of what life can be. Imagine creating entirely new organisms that have never existed on Earth, engineered at the molecular level to perform specific functions. This is not science fiction, it's happening right now. One of the pioneers in this field is the J. Craig Venter Institute, where an ambitious project to synthesize a bacterial genome from scratch significantly advanced our understanding of genetic engineering. In 2010, Venter and his team successfully created a synthetic cell known as Mycoplasma mycoides. This historic breakthrough marked the first time a synthetic genome had been transplanted into a host cell, allowing it to function as a living organism. Such groundbreaking experiments challenge our definitions of life and raise profound questions. What constitutes a living organism? In creating life from scratch, scientists are not only uncovering the fundamental principles of biology but also exploring how we might engineer future organisms for valuable purposes, such as biofuel production or carbon capture. For instance, researchers are developing engineered microbes that convert carbon dioxide into usable fuels or pharmaceuticals. These bioengineering efforts could potentially address the pressing needs of our society while also combating climate change. However, with such power comes great responsibility. The moral dilemmas of creating life forms lead to debates on bioethics. Should we create new microorganisms that could impact public health or ecological balance? The potential for unintended consequences looms large. Synthetic biology could lead to innovations in health through customizable organisms designed to produce vaccines or therapeutics on demand. Yet, the implications of releasing synthetic life into the environment remain unclear. What happens when these engineered organisms interact with natural ecosystems? As we stand at this precipice of possibility, we must question not only our capabilities but also the ethical frameworks that guide us. Can we govern synthetic biology responsibly, ensuring that our innovations benefit humanity without jeopardizing the delicate balance of our ecosystems? The future of synthetic biology depends not only on scientific capability but also on our ability to navigate the ethical landscape that accompanies it. In 2012, a groundbreaking discovery shook the foundation of genetic engineering, CRISPR. This technology, which allows for precise editing of DNA, opened up a world of possibilities. But before delving into its implications, it's crucial to understand what CRISPR is. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's a natural defense mechanism found in bacteria, which they utilize to fend off viral attacks. Through a unique process, bacteria can remember past infections and respond more effectively to repeated threats. In the lab, scientists adapted this bacterial system to manipulate genes in a variety of organisms. The result? A method that is not only accurate but also relatively easy and cost-effective. Compared to older techniques, CRISPR can edit DNA with unprecedented precision. For instance, the first major application of CRISPR took place in mice, where researchers successfully knocked out specific genes linked to certain diseases. But the implications don't stop at basic research. CRISPR has been used to tackle significant challenges in human health. In 2016, a team led by scientists at the University of Pennsylvania conducted a remarkable experiment, they used CRISPR to edit immune cells in patients suffering from aggressive forms of cancer. This therapy, known as CAR-T cell therapy, has since shown promising results, with some patients entering remission. Yet, with this power comes responsibility. The ease of accessing CRISPR technology raises ethical questions. Who has the right to modify a life? There have been controversial cases where researchers have attempted to create modified embryos, raising concerns about the long-term consequences of designer babies. As we dive deeper into the ramifications of CRISPR, we must ask ourselves, should we become architects of our own evolution? The excitement surrounding CRISPR is palpable, but so is the caution it warrants. As this technology continues to evolve, how can we ensure that it is used for good? The next chapter will explore not only synthetic biology but also the critical boundaries we must consider as we redefine our capabilities. Gene therapy represents one of the most revolutionary approaches in modern medicine. Imagine a world where genetic disorders could be corrected at their source. This hope is closer than ever before. In essence, gene therapy involves altering the genes inside a person's cells to treat or prevent diseases. This could mean replacing a mutated gene with a healthy copy or introducing a new gene entirely to help combat a disease. One of the most notable success stories in gene therapy is that of a young boy named Jackson Willard. Born with a rare genetic disorder called spinal muscular atrophy, SMA, Jackson faced a grim prognosis. SMA is caused by a deficiency of a protein that is essential for the survival of motor neurons, leading to severe muscle weakness and, ultimately, death. However, in May 2019, Jackson received a groundbreaking treatment known as Zolgensma, which aims to address the root cause of the disease by delivering a copy of the missing SMN1 gene. As a result, Jackson has shown remarkable improvement in his ability to sit up and even crawl. His story is a beacon of hope, reflecting the monumental potential of gene therapy. But while the success of Zolgensma is thrilling, it also highlights the complexities and costs associated with such advanced treatments. A single dose of Zolgensma can cost over $2 million, leading to questions about accessibility and equity in medicine. As we advance deeper into the world of gene therapy, we also see the promise of treating other diseases like cystic fibrosis, hemophilia, and certain types of cancer. The potential for gene editing technologies like CRISPR to revolutionize gene therapy is immense. For instance, researchers are investigating the possibilities of using CRISPR to correct genetic mutations in patients with cystic fibrosis, allowing for a potential cure rather than the management of symptoms. However, the road ahead is fraught with challenges. Regulatory hurdles, long-term safety, and ethical considerations loom large. The next chapter will shed light on the impact of genetic engineering on biodiversity and the ecosystem, and how these scientific advancements must balance the wonders of innovation with the preservation of our natural world. Have you ever wondered what the future of life could look like if we were able to design organisms from scratch? Synthetic biology brings us at the cusp of this fascinating frontier. Defined as the engineering of biology to create new functions and capabilities, synthetic biology extends beyond mere genetic modification. It encompasses designing and constructing new biological parts, devices, and systems, as well as redesigning existing natural biological systems. One of the most groundbreaking projects in synthetic biology was the creation of synthetic life by a team led by Craig Venter in 2010. They synthesized a bacterial genome, assembling it from scratch and successfully inserting it into a host cell, creating a new organism that was fundamentally different yet capable of living and reproducing. This was a landmark achievement, not only proving that we could create life in the lab but also asking significant questions about the very essence of life itself. Synthetic biology holds incredible promise in various fields, including energy, materials, and medicine. For instance, researchers are engineering microorganisms to produce sustainable biofuels, designed to reduce our reliance on fossil fuels. Imagine if bacteria could consume carbon dioxide and convert it into energy. This is not just a dream but a potential reality on the horizon. However, as we design life, we must also consider the ethical implications. The idea of creating organisms with customized traits leads to profound questions. Should we create new species? What safeguards are necessary to prevent unintended consequences? As synthetic biology continues to progress, it challenges our understanding of what it means to be natural. In the face of these developments, the societal impacts are vast. From the potential to solve global hunger to the environmental sustainability of our planet, synthetic biology could redefine our relationship with life itself. Yet, we must tread carefully, as the next chapter will dive into the complexities of how genetic engineering influences biodiversity and our ecosystems, a balance between innovation and ecological integrity. As we dive deeper into the complexities of our genetic makeup, a fascinating layer of regulation reveals itself: epigenetics. Unlike the sequence of DNA itself, epigenetics involves the chemical modifications that affect gene expression without altering the underlying DNA sequence. This field has exploded in research over the past 2 decades, turning our understanding of genetics upside down. One of the most remarkable aspects of epigenetics is its ability to be influenced by external factors such as environment, diet, and lifestyle. For instance, a landmark study conducted by researchers at the University of California demonstrated that a high-fat diet could alter the epigenetic markers in mice, leading not only to obesity but also to changes that could be inherited by future generations. Moreover, the implications of epigenetics stretch far beyond obesity. In the case of cancer, researchers have identified specific epigenetic changes that can turn normal cells into malignant ones, providing potential targets for new therapeutic strategies. For example, drugs that inhibit the enzymes responsible for adding or removing epigenetic markers are currently in clinical trials. This raises intriguing questions. Can we redefine health and disease through the lens of epigenetic modifications? Further, when we discuss transgenerational epigenetic inheritance, we must grapple with both its promise and peril. Studies involving animal models, like those conducted by researchers from the University of Pennsylvania, show that stress and trauma can leave an epigenetic imprint that can persist in subsequent generations. While this is a powerful mechanism for adaptation, it suggests that our children might inherit the consequences of our life choices, including diet, stress levels, and environmental exposures. Understanding this could revolutionize public health approaches and genetic counseling. We must ask ourselves, how much control do we truly have over our genetic destiny? As we look ahead, the exciting field of epigenetics is opening doors to groundbreaking treatments. Scientists are exploring how manipulating epigenetic markers could provide new ways to treat conditions like schizophrenia, depression, and even aging itself. But with great power comes great responsibility. The implications of harnessing epigenetic mechanisms are immense, and society must weigh the risks versus the potential benefits. The discussion about who gets access to such technologies, the ethical implications of modifying our genetic expression, and the potential for misuse in creating genetic superior individuals will become increasingly critical. One thing is certain: the more we learn about epigenetics, the more we realize just how complex and interconnected our genetic fabric truly is. What does this mean for our understanding of identity, health, and the future of humanity? As we continue to decipher the epigenetic code, we may just find the answers that have eluded us for centuries. Imagine a world where artificial intelligence not only understands our genetic code but also has the ability to manipulate it at lightning speed. The intersection of genetic engineering and artificial intelligence, or AI, opens up unprecedented possibilities and challenges. Today, AI technologies are increasingly being used to predict genetic outcomes, optimize gene editing processes, and even discover new genetic therapies. For example, companies like DeepMind have made significant strides in using AI to predict protein folding, a critical factor in understanding diseases like Alzheimer's and cystic fibrosis. But how does this relate to genetic engineering? By leveraging AI, scientists can analyze vast amounts of genomic data to identify mutations linked to specific conditions. This capability not only enhances diagnostic precision but also accelerates the development of tailored therapies. One noteworthy case involved a collaboration between AI experts and geneticists who devised a model that predicts the efficacy of various CRISPR editing strategies on specific genes. The ability to simulate and visualize complex biological processes in silico allows researchers to test hypotheses faster than ever before. This fusion of disciplines is paving the way toward personalized medicine, where treatments can be customized to an individual's genetic makeup rapidly and accurately. However, the ethical implications of AI in genetic engineering require careful consideration. Questions of access, bias in algorithms, and the potential for unintended consequences loom large. As AI learns from historical data that may contain biases, it could inadvertently reinforce existing health disparities. In addition, the prospect of AI-driven genetic modifications raises significant ethical concerns about accountability and consent. Who is responsible if an AI system suggests a modification that leads to adverse outcomes? Furthermore, the merging of AI and genetic engineering poses the question of how far we should go in modifying life. While AI can help eliminate genetic diseases, the capability to enhance human traits or abilities presents a slippery slope. As we navigate this uncharted territory, we must consider regulations and frameworks that ensure responsible innovation. Looking into the future, as AI technologies improve and become more integrated with genetic engineering, the potential for innovative therapies and enhancements is staggering. We could witness profound changes in fields like agriculture, where AI could create genetically modified crops that are more resistant to climate change and pests. However, are we ready for a world where machines dictate the course of genetic evolution? As we stand on the brink of this new frontier, the convergence of AI and genetic engineering invites us to contemplate our role in the tapestry of life. Will we embrace our newfound powers responsibly, or will we face the consequences of unbridled ambition? Genetic engineering is not merely a scientific endeavor, it is a profound societal shift that carries significant implications for how we view health, identity, and our relationship with nature. As we modify the blueprint of life itself, we must consider how these technologies affect our social fabric. One of the most pressing concerns is the potential for a new form of inequality. If genetic enhancements become accessible only to the wealthy, we risk creating a society where genetic privilege determines opportunities. In this light, the debate surrounding genetic engineering has swiftly evolved into discussions about social justice and equity. Case studies, such as the reactions to genetically modified organisms, GMOs, in agriculture, illustrate the divisions that can arise. Many farmers in developing countries have embraced GMOs as a means to combat hunger and improve crop resilience. Conversely, others argue that such practices erode traditional agricultural systems and can lead to dependency on large corporations. This tension showcases not only the scientific implications but also the profound social dilemmas inherent in adopting genetic technologies. Furthermore, there is the question of public perception surrounding genetic engineering. A survey conducted by the Pew Research Center revealed that while a significant portion of the population sees the potential benefits of genetic modification for disease prevention, there remains a deep-seated fear of playing God. This fear often stems from a lack of understanding of the science involved and the potential risks associated with genetic modifications. Education and transparency are vital as we navigate these waters. Beyond health and agriculture, genetic engineering also challenges our understanding of identity and belonging. As we begin to alter genetic identity, we must consider the implications for personal and cultural narratives. For instance, the use of gene editing technologies might lead to questions about authenticity and what it means to be human. In a world where we can potentially design our offspring, how do we reconcile our different cultural beliefs about family, identity, and the essence of being? This not only extends to individual identity but also to collective histories. The concept of genetic modification leads us to reflect on how much of our identity is inherent and how much is a result of our environment. The complexity of this question underscores the interplay between genetic predispositions and social contexts. Moreover, as gene editing becomes more prevalent, the ethical implications of consent come into play, especially concerning unborn children who cannot voice their desires. As we reshape life, we must tread carefully in defining the rights of individuals, especially those yet to be born. As we look to the future, the ramifications of genetic engineering will undoubtedly ripple through our society in ways we cannot fully predict. Will we embrace our human ability to modify life responsibly, or will we find ourselves grappling with the consequences of a world redefined by our genetic choices? In navigating this new reality, ongoing dialogue between scientists, ethicists, policymakers, and the public will be essential in shaping a future where science and humanity can coexist harmoniously. In the modern age, biotechnology has woven itself tightly into the fabric of our daily lives. From the food we eat to the medicines we trust, genetic engineering has transformed our reality. But what does it mean for society when science transcends natural boundaries? Let's delve deeper into this transformative force. Biotechnology, often described as the use of biological systems or living organisms to develop products, has roots that trace back to ancient times. Humans have been manipulating the genetic makeup of organisms for thousands of years, primarily through selective breeding. But it wasn't until the 20th century that science began to understand the intricate double helix of DNA and how to manipulate it directly. One of the pioneering figures in this field is Paul Berg, who in 1972 developed a method to splice DNA from one organism into another. This groundbreaking work laid the foundation for recombinant DNA technology, which has since evolved into today's sophisticated genetic engineering techniques. Today, genetic engineering technologies enable scientists to not only understand but also manipulate the genetic codes of living organisms at an unprecedented level. As we consider the implications, key areas of innovation include agriculture, pharmaceuticals, and even environmental cleanup. In agriculture, genetically modified organisms, GMOs, have sparked debates about their safety and impact on biodiversity. Crops engineered for pest resistance, such as Bt corn, have reduced pesticide use and increased yield. For instance, a study published in the journal Science reported that Bt cotton and Bt corn reduced pesticide use by over 37%, which has immediate economic benefits for farmers while also reducing environmental impact. Yet, the long-term effects remain a point of contention. Critics argue that GMOs could lead to a decline in biodiversity and create superweeds resistant to herbicides. Furthermore, there's a growing concern about the monopolization of seed markets by a few corporations, raising issues of food sovereignty and security. In medicine, genetic engineering has paved the way for groundbreaking advancements, such as monoclonal antibodies and gene therapies. One striking example is the development of the CAR T-cell therapy, a revolutionary treatment for certain types of blood cancers. This therapy involves modifying a patient's T-cells to better recognize and attack cancer cells. The success rates have been profound. In clinical trials, nearly 83% of patients with acute lymphoblastic leukemia achieved remission. However, the cost of this treatment can exceed $373,000, raising ethical questions about access and equity in healthcare. Biotechnology companies are also turning their sights towards environmental issues. Researchers are utilizing genetically engineered microbes to help clean up oil spills and toxic waste sites. For instance, scientists at the University of California, San Diego, modified bacteria to digest and break down oil spills, effectively turning environmental disasters into opportunities for biological restoration. As we venture further into the age of biotechnology, we must navigate a landscape filled with moral, ethical, and scientific complexities. Genetic engineering offers tremendous promise, but it also demands our responsibility as stewards of the earth. Will we use this knowledge to create a better world, or will we falter in our choices? The implications of genetic engineering stretch beyond science. They weave into the very fabric of society and culture. Decisions made today will impact generations to come, and as citizens of this planet, we must weigh our options carefully. In the next chapter, we will explore the complex relationship between genetic engineering and human identity, focusing on the social and ethical ramifications that challenge our very existence. As genetic engineering continues to shape the world around us, public perception plays an essential role in the trajectory of this science. How do we as a society view these advancements? Are we ready to embrace the potential benefits, or are we tethered by fear and misunderstanding? Public opinion on genetic engineering is complex and often polarized. On one hand, many embrace the potential health benefits and agricultural efficiencies. On the other hand, fears of playing God and unforeseen consequences loom large in public discourse. One notable case is the genetically modified organism debate in Europe. The European Union has stringent regulations on GMOs compared to the United States, where GMOs are widespread and accepted. The cultural context plays a significant role here. In Europe, there's a long-standing history of skepticism towards large corporations and a strong emphasis on food labeling. According to a survey conducted by the European Commission, 74% of Europeans have concerns about the safety of genetically modified food. This fear can be traced to several high-profile controversies, such as the case of genetically modified corn and its perceived role in health issues among livestock and humans. While scientific studies have repeatedly affirmed the safety of GMOs, the media often sensationalizes these cases, leading to greater public anxiety. The social perception of genetic engineering is also colorfully illustrated through the lens of pop culture. Movies like Gattaca and Jurassic Park have often tackled the ethical dilemmas of genetic manipulation. They reflect our profound fears about losing control of technology and the ethical implications of human enhancement. These narratives spark public interest and frame genetic engineering as a dilemma of humanity's future. Furthermore, the ethical implications of genetic engineering extend into the realm of designer babies and human enhancement. The notion of editing genes to prevent diseases or enhance traits raises questions about identity, diversity, and societal expectations. In 2018, the world was shaken by the news that a scientist in China, He Jiankui, claimed to have created genetically edited twins resistant to HIV. This revelation sparked international outrage and discussions about ethics in genetic research. The ethical overlap with genetic engineering and social equity is profound. If genetic enhancements become commercially available, will they be accessible to all or just the affluent? This could exacerbate existing social inequalities and lead to a bifurcated society of enhanced and non-enhanced individuals. It poses questions about what it means to be human and how much control we ought to exert over our biology. As we navigate through this evolving landscape, education becomes paramount. By fostering a better understanding of genetic engineering, its benefits, and its potential pitfalls, we can help illuminate the path forward.